Okay, class, let's keep moving and shaking through this chapter on blood vessels. We left off talking about the differences between arteries and veins. Now we're going to start talking about capillaries and switcheroo right here. And section 18.3 if you're following along in the Harvard Community College textbook. So, typical arteries. I mentioned before that not all capillaries are the same. So a typical capillary has just a tube of endothelial cells. Picture like just a cell tube, one cell thick is all it is. Very delicate basement membrane, no tunica media, no tunica externa. So no muscular layer, no connective tissue layer. Very small. It's about the same diameter as a typical red blood cell, which is nice because those red blood cells stack up in the rouleau so they can squeeze through the capillaries, right? There are two major types of capillaries. You have continuous capillaries and fenestrated capillaries. So continuous capillaries are ones where there's a complete endothelial lining. Again, the tube, the cell tube. Think of that. This is the majority of the body, except our epithelial and the cartilage. This is where water can diffuse, small solutes, lipid soluble materials, all can just squeeze right on through those cells and get to where they need to go. Some specialized continuous capillaries are in the central nervous system and in the thymus that have very tight junctions between those endothelial cells to restrict the permeability and only let certain things pass. So they are even more limited in what they allow through. So again, in order to get from one side of the capillary to the other, the stuff, right, the oxygen or water or whatever, it literally has to pass through the cell. So it's in the blood, it has to go into the cell membrane of our endothelial cell and then out the other side to get to the interstitial fluid, right? We're not talking about squeezing through the gaps between the cells in a continuous capillary. In a continuous capillary, in order for stuff to get from the inside of our blood to the interstitial fluid or from the interstitial fluid to the blood, it has to go through that endothelial cell. Seems like a lot of work, right? So some capillaries are even more specialized and they're fenestrated, or they have windows on them. These windows are basically pores between the endothelial lining. So this is going to allow for even more rapid exchange of water, solutes, etc. So this is probably what you thought capillaries were. Instead of the substances, like in a continuous capillary, having to go from the blood across the plasma membrane of the endothelial cell, through the cell, and then across the plasma membrane again to get to the interstitial fluid. And fenestrated, there's these little tiny pores in between those endothelial cells to allow for exchange of materials. These occur in the choroid plexus of the brain. Pop quiz, what the choroid plexus do? Make CSF, right? So choroid plexus of the brain, where one of these sites of fenestrated capillaries occur. Hypothalamus, pituitary, pineal, and thyroid glands. That makes sense, right? What are those things doing? Secreting hormones, right? You got to get those big hormone protein molecules into the blood. They're also in the absorptive areas of the intestinal tract. Makes sense. You need to get all those nutrients in. And certain sites along the kidney for filtration. So here you see the big difference between a continuous capillary and a fenestrated capillary. The fenestrations, or the windows, allowing for rapid exchange of solutes. The continuous is just a little bit more of a lengthy process because the substances have to hop through those endothelial cells. There's also sinusoids. Sinusoids are kind of like a more elaborate version of the fenestrated capillaries. They're more flattened and irregular. They have gaps between the endothelial cells. The basement membrane is almost gone, non-existent. This is for water and solutes, even plasma proteins to be exchanged, so even larger substances. These are located in the liver, the bone marrow, the spleen, and several of our endocrine organs. So if we think of the functions of these organs, the liver is making plasma proteins, so those plasma proteins obviously need to exchange. The bone marrow is secreting blood cells, those stem cells that need to go out into the world of our body, right? The spleen, which is absorbing our red blood cells and recycling them, and our endocrine organs, again, making those big hormone molecules, which are proteins usually, that need to get into the blood. So those 4 or 5 main sites need these sinusoids in order for these proteins to be exchanged. So there you can see sinusoids with these little slits almost through them. So our capillaries are arranged in what we call capillary bed. It's basically an interconnected network of all of these little cell tubes connecting together. So there's several connections between arterioles and venules. So it's not just an arteriole leading to one tube then going to a venule. It's a whole network. Think of like a tree trunk, right? You have this big trunk that branches literally into branches, and then smaller branches, and then twigs, right? And then if you imagine that tree trunk being attached, that tree being attached to another tree via those little twigs, it's kind of like what these capillary beds are. They're the twigs of the trees that are touching. So they can be supplied by more than one artery, and they're called collaterals, which if a bunch of arteries lead to the same capillary bed, it's called a collateral. They can fuse before giving rise to the arterioles, and fusion is an example of what we call an arterial anastomosis. An anastomosis is the joining of blood vessels. So if one artery is blocked, another artery feeds that same capillary bed, so the effects won't be as bad if one of the arteries is blocked. It's kind of like a backup plan, right? So this is going to allow for a continuous flow of blood through the area. The capillary bed can also be bypassed, an arteriovenous anastomosis. So this is where the arteriole is directly connected to the venule. In other words, no exchange can take place. This can be controlled through our nervous system. Sympathetic innervation can block off a capillary bed and redirect the blood somewhere else. This happens during sympathetic innervation in our digestive tract, right? Sympathetic is fight or flight. We don't need to digest that cheeseburger that we ate earlier. We need to activate our heart and muscles so that we can get the heck out of this dangerous situation. Makes sense. There's also a meta-arteriole or a pre-capillary arteriole. This is the initial segment of that connection pathway, and this is where there's a smooth muscle that can actually change the diameter or flow rate of that capillary bed. So it's kind of adjusting the flow. Our capillary bed can have a couple different properties, a thoroughfare channel, and you're going to see a picture of this on the next slide, or if you're super smart, maybe you have this PowerPoint printed out already, and you can actually look at that slide as I'm talking. That might help you. So the thoroughfare channel is the most direct pathway through a capillary bed. It's literally arterial, capillary, venule. There's no other like flowery extra stuff. There's precapillary sphincters that can be present. Precapillary sphincters are bands of smooth muscle on the arterial side which can open and close to allow blood to flow through or to block blood flow through a particular channel. Vasomotion is this cycle of contraction and relaxation of these precapillary sphincters. So let me flip ahead here, and you can see how this might occur. Here's the arterial side, our blood. First, we have arterial collaterals, which are fusing in an arterial anastomosis. The anastomosis, which leads to our arteriole here. Arterioles are going to kind of branch off into different thoroughfares, different pathways, and these pathways are controlled by precapillary sphincters, literally little circular muscles that are at the opening of all of these capillary beds, at the arterial side of all of these capillary beds. So you can imagine each of them kind of opening and closing at different rates to direct the flow of blood, because in theory, if this first channel was open all the time, the majority of the blood will flow through that first channel, right? And then only a trickle would get down here. So your precapillary sphincters are opening and closing to direct that blood flow to each of the different passageways so that blood can flow evenly through the tissue. The thoroughfare channel you see here leads eventually to our venous side, where we have our small venules leading to our larger venule, which fuses into the medium veins, which fuse into the large veins, which dump back into the heart, right? So that's the whole passageway. That's the whole sequence of events. You can pause the video here, go back and answer these questions. Our next video is going to get more into the physiology piece of this chapter. So let me know if you have any questions. Bye.